Good morning. In this session, we are going to investigate the factors necessary for photosynthesis. That is, we are going to analyze the results of specific experiments, which were performed in order to determine the factors required for photosynthesis to take place in green plants. Of course, we should follow the steps of the scientific method, in order to reach at a conclusion. In this example, the man has observed that the growth rate of plants varies depending on the type of fertilizer he added to the soil. A question arises, and he wanted to know, which fertilizer is the best for plant growth. In his opinion, fertilizer A is the best one to be used, but he was not sure. That's why, he decided to perform an experiment in order to test if his hypothesis is valid or not. So, he experimented on 3 plants, which were of the same species and of the same starting size. The first plant was grown in the presence of fertilizer A, the second plant was grown in the presence of fertilizer B, and the third plant was grown in the presence of fertilizer C. After that, he kept the 3 plants in open air, in the presence of sunlight, and of course, he watered them regularly. 10 days later, he measured their size, and inspected their health state. He compared the results and he noticed that the highest growth rate was in the first plant, which was grown in the presence of fertilizer A. This analysis led him to a final conclusion: fertilizer A is the best fertilizer for enhancing plant growth. Of course, in this case, his hypothesis was valid. So, for example, if we want to determine the factors essential for photosynthesis, there are certain important points we have to take into consideration before designing an experiment. First, identifying the studied problem. Example, is light essential for photosynthesis to take place? And of course, formulating a hypothesis. Then, specifying the independent variable, the variable factor that I have to change in order to study its effect. Including a control for comparing results. Using techniques that allow us to observe and measure results. And finally, analyzing the obtained results to reach a conclusion, which is an answer to the posed problem. We already know that photosynthesis is a biological process that takes place in plants, to synthesize organic food substances, mainly starch, and this process is essential for plants' survival. In order to identify the factors required for photosynthesis and to determine their effect on the photosynthesis rate, different experiments will be performed and we are going to analyze the obtained results to reach an adequate conclusion. The first experiment is done to study the importance of light. 2 plants are grown in wet soil rich in minerals, such that plant A is covered with a transparent bag allowing the sunlight to reach the plant. Plant B is covered with an opaque bag preventing the sunlight to reach the plant. Before looking at the obtained results, let us answer some questions. Indicate the variable factor, that is the independent factor that is under study. Pose the studied problem, that is the question that we are trying to answer using this experiment. Formulate a hypothesis, meaning, give a hypothetical answer to the posed problem. We will see later if your hypothesis is valid or not. Pause the video, and write your answer, then we will correct them. Now let us see the correct answers. Both plants were kept under similar conditions, but the only different condition was light. We have seen that plant A was kept in light, while plant B was deprived of light. Thus, the independent variable or the variable factor is the presence and absence of light. Concerning the posed problem, this experiment was done to study the effect of light, that is to see if light is essential for its growth and therefore is essential for photosynthesis. So, the problem under investigation. Is light essential for photosynthesis to occur? Well, it might be essential, as I think, and so, this would be my hypothesis. Light is an essential factor for photosynthesis. Now, after leaving the plants in such conditions for few days, we got the following results. Plant A survived, while plant B died. Now, let us interpret the results. Meaning to see what the results prove. And then, let us think, what would be the answer to the posed problem based on our analysis? Of course, our conclusion would be the answer to the posed problem. First, we have to find out the relation between the variable factor and the results. In the presence of light, plant A survived, but plant B died in the absence of light. This is a proof that light is essential for plant survival. Moreover, we have seen that photosynthesis and plant survival are linked, meaning that, if the plant is not able to undergo photosynthesis, it would die. So, in this case, plant A was able to undergo photosynthesis due to its exposure to light, while plant B could not undergo photosynthesis due to the absence of light. So, we were able to prove that light is an indispensable factor for photosynthesis. Read the answer, and let us see what should be included and why. According to the Lebanese curriculum, interpret means, what do the obtained results prove? That's why, the answer should include analysis followed by inadequate significance. The analysis should include the conditions, mainly the variable factor, as well as the obtained results. Note that the difference in condition is responsible for any difference in the result between the experimental setup and the control. Of course, we should use comparative connectors to show this difference. In this answer, I have used the term while, but you can replace it by however, on the other hand, but, and so on. The significance should include the relation between the variable factor and the results. In this case, the relation is between light and plant survival. So, we have proven that light is essential for plants' survival. Concerning the conclusion, the plant survival was used as an indicator of photosynthesis. And, yes, we have proven that light is essential for photosynthesis, since the plant needed light to undergo photosynthesis and survive. So, the conclusion would be that light is an essential factor for photosynthesis in plants, and this would be an answer to our posed problem. Let us watch this experiment, which was also done to help us find out the importance of light for photosynthesis in plants.
To demonstrate that light is necessary for photosynthesis. Procedure. Take a healthy potted plant and place it in dark for about 72 hours so that the leaves become free from starch. After 72 hours, select a leaf on the plant and cover a portion of it on both sides with black paper. Keep the potted plant with the covered leaf in sunlight for a few hours. After a few hours, detach the covered leaf from the plant and remove the black papers. Boil the leaf in water for a few minutes. Now boil the leaf in alcohol in a water bath till it becomes dull white due to the removal of chlorophyll. Wash the leaf with water and add iodine solution. Observation. The portion of leaf which was covered does not change color, whereas the portion which was uncovered turns blue-black. Inference and conclusion. The portion of leaf which was not covered received all the factors necessary for photosynthesis, Therefore, that part could only photosynthesize, whereas the one which was covered could not manufacture food. The presence of starch gave the positive test with iodine, that is, turned to blue-black color. Photosynthesis is the process of manufacturing of food by the green parts of the plant, especially the leaves, in the presence of sunlight and chlorophyll, with the help of carbon dioxide and water.
So far, we know that light is essential for photosynthesis, but what about the effect of light intensity on the reaction rate? In order to answer this question, a plant was placed in different light intensities while keeping all the other factors constant, then the volume of oxygen released by photosynthesis was measured. Note that photosynthesis is a biological process and we cannot observe this phenomenon directly, but we can study certain factors that can act as indicators for its occurrence. In this case, we will measure the volume of oxygen released, since oxygen is a product of photosynthesis, and if oxygen is released by the plant, this means that the plant is undergoing photosynthesis. Now let us look at the recorded results, which are presented in this graph. You can see at this point, where the light intensity is 0, the volume of oxygen released is 0, meaning that in the absence of light, oxygen is not released. Then you notice that, in the presence of light, that is for light intensity greater than 0, oxygen is released, and the volume of oxygen released depends on the light intensity, such that it increases till reaching its maximum, which is in this case, at light intensity of 30,000 lux. But if the light intensity is higher than that, the volume of oxygen release decreases, and you can see the rapid decrease in oxygen release, until it is no longer produced, at light intensity of 33,000 lux. So, we can say, that oxygen is not released in two conditions. First, in the absence of light, and second, if the light intensity is higher than a certain limit. Before going into more details, let us answer some basic questions. Pose the studied problem. And indicate the variable factor. We will see the answer in the next slide. The posed problem for such an experiment would have been: what is the effect of light intensity on photosynthesis rate? Or is photosynthesis enhanced by higher light intensity? And the variable factor in this case would be the absence and presence of light, as well as the variation in the light intensity. To interpret the graph, we have to analyze the obtained results, to reach at a significance. We will start with the first point. In the absence of light, oxygen is not released. But then the results changed, when light is present. So we say, however, in the presence of light, and as the light intensity increases to 30,000 lux, oxygen is released and the released volume increased to reach a maximum of 60 arbitrary units. But then, there is also a change in the results, as the light intensity increased more than 30,000 lux. So we say, whereas, as the light intensity increases furthermore, the volume of oxygen produced rapidly decreases to become null, at light intensity of 33,000 lux. Of course, such results proves two things. First, light is essential for oxygen release by the plant. Second, variation in light intensity enhances the release of oxygen but within a certain limit, such that beyond 33,000 lux, oxygen release is inhibited. Of course, the volume of oxygen release is related to the rate of photosynthesis. That is, if oxygen is not released, this means that photosynthesis is not taking place, but when oxygen is released, this means that photosynthesis is occurring and the volume of oxygen released increases with the increase in photosynthesis rate. Therefore, our conclusion would be: light is indispensable for photosynthesis and it enhances photosynthesis rate within a certain intensity limit. Now, how can you explain the absence of photosynthesis at light intensity greater than 33,000 lux? Formulate a hypothesis. It could be that the high light intensity destroys the chlorophyll molecules, which are essential for light absorption required for photosynthesis. In this session, we have studied the importance of light for photosynthesis and the effect of light intensity on photosynthesis rate. But what about carbon dioxide? We will see in the next session. Goodbye for now and thank you.